this lecture is about the uric acid formation by the catabolism of purine nucleotides. As the purine nucleotides are the adenine and guanine. So adenine and guanine are the purine bases that we get through the food. So after getting the food sources that are rich in these purine nucleotides, the food they pass through the after taken up by the human, then they pass through the digestion processes, and after digestion they are absorbed into the blood circulation. Through blood they reach to the liver. So the catabolism of these purine nucleotides takes place inside the liver, and in the liver the uric acid formation takes place. So the end product of these purine nucleotides, adenine and guanine, is the uric acid. The uric acid is synthesized in the liver and then eliminated in the body through the kidneys. So these uric acid are soluble, so they are easily excreted and eliminated from the body through kidneys. So you can say that the oxidation of the purine nucleotides, adenine and guanine, is the end product of the purine nucleotide bases. Adenine and guanine is the uric acid. So then uric acid, they are less toxic and they are eliminated from the body. Okay. So for the synthesis of uric acid, as the purine nucleotides are the adenine and guanine. So adenine and guanine are present in the body in the form of adenine monophosphate and guanine monophosphate. So these adenine monophosphate and guanine monophosphate, these are the parent nucleotides that are synthesized as guanine monophosphate. It is synthesized in the body through two pathways. There are two synthetic pathways for the synthesis of the guanine monophosphate, guanosine monophosphate. As these nucleotides are taken up through food, and then there are numerous enzymes that act on these polynucleotides, and then after uh, after passing through the digestion and absorption processes, then there are some synthetic processes. That through synthetic processes, there are two types of synthetic processes. There is a de novo synthetic pathway and salvage synthetic pathway. So through that pathway, guanine monophosphate and adenine monophosphate is formed. So guanine monophosphate, it passed through a catabolic pathway for the formation of the uric acid. So uric acid is synthesized from the guanine monophosphate. It passed through a series of reactions. So there are almost four reaction pathways. There are four reactions through which this guanine monophosphate is converted into the uric acid. So let's see each reaction in detail. First of all, the guanine monophosphate is converted into the guanosine. So this is the structure of the guanosine monophosphate. As the guanosine monophosphate, as the name indicates, there is the guanine nucleotide base, attached to the ribose sugar, and there is the phosphate. So this is the guanosine monophosphate. So guanosine monophosphate, because there is the monophosphate, it means that there is single phosphate attached to this guanosine. So guanosine, this is guanine base attached to the ribose, so this is known as guanosine, and there is the phosphate attached there, so it is known as guanosine monophosphate. The enzyme nucleotidase in the presence of water molecule. Then what will happen? Then it will release this water molecule, will act on this, will break this bond between the phosphate group and the ribose sugar. So the bond between this phosphate group and ribose sugar is broken down by this water molecule with the help of the enzyme nucleotidase. It will set free this nucleoside free. So this nucleoside, this guanine nucleotide and ribose will be separated, will form guanosine. This guanosine together with phosphate is known as guanosine monophosphate because it's a single phosphate. So when the phosphate is removed, this guanosine monophosphate will become the guanosine. It's the guanine attached with the ribose sugar, and the phosphate group is released with the help of this water molecule and this nucleotidase. So nucleotidase will convert this guanosine monophosphate into the guanosine through the removal of this phosphate from this GMP. Okay. Now in the next reaction, this is the formation of the guanosine from the guanosine monophosphate with the help of the enzyme nucleotidase, that the water molecule, in the presence of the water molecule, this phosphate is released. Okay, then in the next reaction, this guanosine, in the second reaction, the guanosine is converted into the guanine. So let's see this reaction in detail. That guanosine is converted into the guanine. That guanosine is the guanine base attached with the ribose sugar. So the enzyme act will be the nucleoside phosphorylase. That in the presence, this nucleoside phosphorylase enzyme will act in the presence of the phosphate it will remove this ribose from this guanine and will set this guanine free. Okay. So this nucleoside phosphorylase, in the presence of phosphate, this phosphate will attach to, this is the structure of the ribose sugar. This phosphate will attach to the carbon number 1 of this ribose sugar. Then it will become ribose 1 phosphate, because this phosphate will attach to the first carbon of this ribose sugar. Then this ribose 1 phosphate will be free and this guanine will be free. Then if this nucleoside phosphorylase enzyme, it cleaves the bond between this guanine and this ribose sugar with the help of this phosphate group. So this phosphate is attached to the first carbon of this ribose sugar, then it will become ribose 1 phosphate, and then this guanine is set free. So guanosine is converted into guanine with the help of the enzyme nucleoside phosphorylase. In the next reaction, this guanine is converted into the xanthine with the help of the enzyme guanase. As this guanine, as you can see in this reaction, in the guanine there is the amino group attached here, and then in the xanthine, this amino group is replaced with the oxygen group. So you can say there is a deamination, this amino group is removed and replaced with the oxygen group. So guanine is attached with the xanthine. Xanthine and guanine have the same structure, the same, but the difference is that this amino group is replaced with the oxygen group. So guanine is converted into xanthine with the help of the enzyme known as guanase. Now this xanthine again is converted into the uric acid. Now finally there is the formation of the uric acid, as uric acid is the end product of the uh, guanine catabolism. So this xanthine is converted into the uric acid with the help of the enzyme known as xanthine oxidase. So this xanthine oxidase, so it means it's going to oxidize this, cause the oxidation of the xanthine. So in this reaction, it also required the presence of the water and oxygen, and this water and oxygen is converted into hydrogen peroxide. So there is the formation of the hydrogen peroxide and uric acid in this reaction. So this xanthine, as you can see in the structure here, the oxygen, additional oxygen group is attached. There are 2 oxygen groups are attached, but in uric acid there are additional 3 oxygen groups are attached. So this xanthine oxidase is going to attach the oxygen, it attaches the additional oxygen group to this xanthine group and it becomes the uric acid. So in this reaction, there are the two end products, uric acid and hydrogen peroxide. So this hydrogen peroxide is then again catalyzed by the enzyme known as catalase and will convert this hydrogen peroxide into the water and oxygen. So now the xanthine, in the last reaction of this guanosine monophosphate catabolism, xanthine oxidase converts the xanthine into uric acid. So this uric acid is then soluble substance and then they are eliminated through the kidneys. So this is the overall process that we have seen each reaction in detail. That for the guanine monophosphate, as the guanine monophosphate, it is synthesized in the body through two synthetic pathways. It's the gene-over synthetic pathway and salvage synthetic pathway. And this nucleotide catabolism or oxidation occurs through a series of reactions and is finally oxidized into the uric acid. And then uric acid is excreted from the human body through urine. So guanine monophosphate is a 4-step reaction, and in this 4-step reaction, different enzymes are used, in which the guanine monophosphate is first converted into the guanosine. As the guanosine monophosphate, the phosphate is removed and to form the guanosine, and then from the guanosine, the ribose sugar is removed, then it will become the guanine. Then the guanine is converted into the xanthine, and then from the xanthine, there is the formation of the uric acid.